Well, good morning. Thank you for having me here today. This is a very big shift in gears from what we've been hearing this morning, but prelude to what will come next. And I'm going to talk to you about breeding strawberries for enhanced ornamental value. I just recently transitioned to postdoc, and I'm with the Tom Davis Lab. So my interest started in wanting to breed strawberries with red flower petals, and then also to then breed them for good fruit quality and day neutrality. Strawberry production is quite big internationally at 10.2 billion pounds and 1.1 million pounds in New Hampshire. And the average per capita consumption is 9 pounds a year. So we consume quite a bit of strawberries, and probably these numbers are understated. Strawberry is a member of the Rosaceae family, as are peach and almonds and roses. Strawberries are genetically complex. They have 8 sets of chromosomes, which makes the next part of my talk, as you'll see, more complicated, because as you know, we humans have only 2 sets of chromosomes, and now we have 8 sets to deal with. The ancestors of the strawberry are 2 wild species, Fragaria chiloensis from South America and Fragaria virginiana from North America. And whoa, I didn't mean to do that. The one that was cultivated or crossed to produce Fragaria ananassa was a white fruited type, and the one that I've been working with is a red fruited type. I'm dividing my talk into really 2 sections. One of pre-breeding, which was focusing on getting the red flower color and starting to get some good fruit quality and day neutrality, and then integrating that with cultivars to develop some varieties. And I'm using traditional breeding. This is not anything about GMOs. It's totally traditional breeding, but with a genetic component to help in the breeding effort. The breeding that I'm conducting is at the McFarland greenhouses at UNH and at the Woodman farm, where some test plots, also in Durham. This shows a pedigree of 4 generations in which I was able to obtain the red flower color, and this is the distribution of flower color within each of the generations. I don't expect you to be able to see all of these, but just gives you an idea of, like in this family, 1/4 of them were white, and then in this family, none of them were white. And these are the parents that I utilized to intercross with each other to obtain this red flower color. And I had one that was pink flowered. In this 4th generation, this is where I saw a large emergence of diversity, as I say here, with real deep scarlet flowers and lavender, and some colored on the radius, and some with anthers, which are the pollen-producing parts, and some without anthers. Now, in strawberry, in both the flowers and in the fruit, the pigment is the result of anthocyanins, primarily 2 anthocyanins, pelargonidin and cyanidin. Pelargonidin imparts the orangish color, and cyanidin imparts the strawberry type color. And when you have just cyanidin in the flower petals, you see pink flowers. But with the introduction of the pelargonidin, we have red flowers. It impacts the hue. Now a little bit about the fruit quality improvements. In the initial, I don't know if this is working. I guess it's not. In the initial generations, first through the fourth generations, well, these are the founders in the first three. And you can see that, I'm sorry, the founders are here. And they have very small, pretty inconspicuous fruit. And then as we move through the generations, through the third, it's still not so good. But now in the fourth, it's starting to get better. And now this is introgressing a red-flowered variety with a cultivar. And the fruit is resembling cultivars are fruit that you might purchase at Hannaford's. And again, this is a picture of one of my advanced selections that has the red flower color and its fruit. And this was the first occurrence of red flower color. Within the strawberry breeding, we have a large strawberry community and Rosaceae community that helps provide us with tools to advance the next part, which is marker-assisted breeding, and has recently been funded for another 5 years. And we have meetings every 4 years internationally with strawberry, where we meet with all kinds of other people who are interested in strawberries. As part of this, we, in conjunction with this Rosaceae community, developed an array that can scan the genome of strawberry at over 90,000 different sites. With that, we can analyze the data and see if we can find a trait marker association. And so a marker is where the DNA might be different at one spot, and that one spot would correspond to a trait of interest. And for instance here, the DNA might have 2 Gs, which correlates to white, in our case, or it might have 2 Ts, which correlates to color, and a TG would also correlate to color, in a background of Gs, which is the 8 sets of chromosomes that I was telling you about. And so that T would be predictive of color. This is a rather simplified example, but just to give you an idea. Whoops. Oh, what did I do? Okay, good. So as I was telling you, these chromosomes here are predictive of color. So to summarize, we have a diversity of flower color now. We have the red flower color and fruit quality combined. And we're looking for further marker trait associations in addition to color. 10 of the hybrids have been selected for a variety evaluation. And next, I'm hoping to develop new varieties for New England production and Northeast production to include disease resistance, which would be our next step in marker-assisted selection, and short day and day neutral varieties. And I'd like to thank the Agricultural Experiment Station and all those who've helped to advance this effort. And do you have any questions?
So what sorts of disease traits are you going after?
Well, root pathogens, basically. There's a complex of root pathogens that we're really developing some information on. We'll be discussing this at our meeting next week, actually, in Michigan, at our rose breed meeting.
So do you have a yearly rose breed meeting in the States?
Yes, at Michigan State University. Yep.
And what type of ornamentals? Are you working with any local growers, or?
I am. I'm working, well, with Pleasant View Farms and Proven Winners. They are the ones that have identified these 10 varieties they'd like to trial.